Our next speaker, it's Silvia Barbaresi. She's a co-founder and chief scientific officer at Minutia.AI. Her topic is Minutia.AI. Hope I pronounce it correctly. Advancing gut microbiota analysis for healthy aging and disease prevention. So Silvia, we are ready to listen to you.
Yes, good afternoon. Thanks for inviting me. I'm sharing my screen. Hopefully it works.
Yep. Go ahead.
All right, so thanks for having me. I'm Italian. I'm a sports scientist from Ghent University. I studied also for a period of time during my PhD at the Aspire Academy for Sports Science in Doha. It's quite familiar with that region. By the way, we are new in Dubai. Minutia.AI is a startup already based in Singapore. We are a team of different background and scientists, also bioinformatics, and me, I'm apporting information as nutritionist, sports scientist, and biotechnology as well. Our focus, of course, is gut microbiota analysis, and we also apply our own models, artificial intelligence models. So I will go through this very quickly since we already touched on those topics with the two previous speakers. We already know more or less that we have some hallmarks of aging and they are already consolidated in time and science. We know that we can move from genome instability, telomerase, attrition or exhaustion. We can have epigenetic as well, loss of proteostasis, problem with nutrient sensing, mitochondrial dysfunctions, very important, the concept of mitomesis in longevity, cellular senescence, and this is also quite tricky. We know that there are some senescence-associated phenotypes, some secretory pattern that can interact with the gut microbiome and the leaky gut, stem cell exhaustion, and also different intercellular communication, some crosstalk that can be problematic. We know sometimes we heard about those topics for sure, and we also know that there are works ongoing, some eventually interventions, some disease correctly designed to fix those problems. By the way, the problems still remain. The question, why do we age, still remain unanswered. And simply, according to me, my personal opinion, we have to accept the concept of aging as part of life. We cannot reach immortality. What we can find eventually is premature aging or disease associated with aging or inflammation associated with aging. That is completely another kind of problem. And this is the direction where we are going. So we have different theories. We can more or less agree with one or the other, from evolutionary theory or biochemical theory, neuroendocrine, immunology theory. By the way, our focus is this. Actually, we think that attention should be paid on the interaction across gut microbiota and host, in the gut of the host in this case. As Paulina said before, we have different kind of microbiota across the body, but this interaction is really crucial because we have trillions of cells sitting here above the mucus layer, continuously interacting and releases molecules that are captured by our epithelial barrier, our gut cells. And this continuous interaction can be sometimes positive or negative according to the composition that our gut microbiota has. This is the big picture. We can really summarize the complexity of the topic with this picture, but this is, of course, not exhaustive according to me, so it's just touch base of the topic. We know that we have, with different colors, we can identify different kind of human microbiota. So it's not just the gut and stool that is in light blue, but we also have a skin, tongue, so the mouth microbiota, the oral microbiota that is very connected to our lung microbiota, nose, vagina, ears. We have microbiota everywhere, and we are also aware that we have several axes across our body. Focusing on the gut, we know that we have mainly 6 phyla. and different kinds of genus and species. This is more or less the picture of a core microbiota, but we also know that we are very different one to another. We can say that gut microbiota is like a visceral fingerprinting, so we are all different according to our composition of the gut microbiota. We cannot replicate it, so it's very important because we are actually keeping our individuality with this microbiota. We have so many genes in this consortium that we call altogether microbiome compared to the human genome that is very poor if we see the numbers. And we see also that the difference is much higher in this scenario of microbial genome compared to the human genome. So just taking in mind and fixing on these numbers, it's very crucial to consider the importance gut microbiota has on our health. And we for sure cannot prescind from touching this topic in a talk of longevity or general health. So this is something that has already been mentioned, but complexity, according to us, it's very high to be simplified in one slide. So we should talk hours maybe to unravel the topic. But we see that there are axes across the body with every organ, every tissue. In this picture, for example, the gut muscle axis is not mentioned, the gut lung axis. Last year on Cells appeared a paper with the gut highs axis. So actually we have so many axes that I call highways in our body, where indeed microbes are interacting with the host, secreting molecules, releasing different kind of residues from their metabolism, working on our metabolism and releasing subproducts. In some way, they can be pro-inflammatory. In other ways, they can reduce inflammation. All depends on the composition of that our microbiota have. So we try to create our own models. We have our own unit on research and development of bioinformatics, and we try to simulate the interactions, not just across the microbes in the gut, but also with the host. So this is something that for sure we mentioned, but the topic is rather new. So we are aware that the microbiome is doing something and that is really affecting our health and longevity, but maybe we don't know exactly how to use this tool and what to do with this information. So it requires a lot of knowledge just to understand what's going on in the microbiome, but then we have to be able to integrate this output from the microbiota test with the host biomarkers and the host information. So I saw recently a review that is touching nicely the topic, and something is going in this direction. We are exactly aligned with this work to integrate information from the host affecting longevity, like inflammaging, epigenetic, transcriptomic, metabolomic, as we said before, all the omics, with the microbiome omics. That we can define gut clocks. In this way, we can work integrating the information in a predictive way and on the concept of biological aging, not just chronological aging. And this is our proposal of the workflow. Once we gather information through some very deep gut microbiota testings, like deep shotgun metagenomics or whole genomic sequencing, we retrieve information not just on the taxonomy, but also on the functional approach of the gut microbiota. Across different stages of life, we have different population because microbiota is following different trajectories. So maybe it's too late when we want to work to fix longevity on elderly people. Maybe we have to think about doing something before, when we are in adulthood or eventually in our youth, because gut microbiota can be shaped. This is the good news. It can be shaped with lifestyle, exercise, nutrition, also with drugs, also with sometimes epigenetics. So we can do something, but we have to know what to do. So we integrated this information. We applied our model also in geroscience. And we release what we call digital twins, not just in longevity models, but also in sport for optimal performance, fixing injuries in athletes, and also some models for disease prevention. We also have Minutia Singapore working on infant nutrition, studying all the stool residues of newborn to see if we have to do some precision nutrition intervention in them on their early life, early months. During the weaning with some integration. What we do exactly is retrieving information, not just from the gut, but also from the host, and putting all this information together that we release in a report that is very comprehensive. From the host, we normally retrieve information like body composition, inflammatory markers for blood, urine, saliva, their current nutrition. We have also a panel of polymorphisms, genetic polymorphisms, epigenetic when it's possible, and then some performance measurement when we have to deal with the athletes. And then we fix all this information in a model that is predictive and that we call digital twin, that has to be reached across several tests, retests, and then can be validated and inquired to see if we can do something in our lifestyle or nutrition to prevent disease or prevent, as I said, premature aging or injuries if we are especially focused on sport. Digital twins are not our invention, of course, and this is something that already has been done since times of studies and can be applied to different models. The infant microbiome, it's something we're already doing in Singapore, but we can also use this technology for longevity and sport, as I said. It's not based on single biomarkers. This is the real news, but what digital twins are instead are real projection, we can say in silico creation of the gut of the person and eventually of the person on the wall. And we simulate the interaction, continuously interaction that there are across host and microbiome, considering also the physiology, the immunology, and the nutritional conditions of the host itself. This is the way we try to unravel this complexity, but as I said, we need a lot of data and metadata to work on this model properly validate. And digital twin is not a technology just to plug and in. We need to test and retest for having a digital twin of one person at least 3-4 times the gut microbiome during 1 year to see the evolution and then correlate with proper host biomarkers and physiology, immunology, and nutritional other information. This is a mechanic framework, and this is something very interesting because it has a probabilistic and also causality approach. We can eventually predict something and try to understand the cause-effect connection, since we are not just working on individual biomarker on individual bacteria, but we are recreating the net itself. And this is real an advantage, but this is something that takes time, it's time expanding, and also has its own costs. So this is also a simulation of our models. For example, here we were working on a football player. We used several markers, and we were trying to create connection and see what was going on, changing one values on the others. So we were making a mock-up experiment. We also can use the gut microbiome to calculate some index, gut microbiome health index. This is something we are already doing. In the end, we want to address this question, giving your age, your lifestyle, your current nutrition, your physical status, and your microbiome status, which is the risk that you are going to suffer from a disease, or eventually how can we manage your health to improve your performance, or what can we do to prevent a sort of premature aging or inflammatory pattern that is already ongoing. So what can we do to reverse this situation? So just to wrap up my presentation, this is something that we normally provide to our client to understand our models. So we have explainability for sure, because we want to unravel the complexity of these biological mechanisms. We have cause-effect reasoning, of course, because it's a mechanicistic models that is creating the whole interaction on a mathematical framework. We have also ability to create integration with the host data. We try to fix what's the problem on an uncertainty, and also temporal dynamics. This is important because, as I said, we cannot even rely on one single test to have a digital projection, but we need to test, retest people at least 3-4 times before having the whole picture and having a digital twin technology well validated. So I conclude my presentation. I hope to have some questions to answer. And again, I thank you for your attention and the opportunity to join you today.
Thank you so much, Silvia. It was very insightful, and I was very curious about the practical applications of the longevity for today, especially about the digital twins and sports. Thank you so much.